Breaking news. Those Thanksgiving brownies may alter the community of microorganisms inside your mouth. The ecosystem of microorganisms that inhabit the mouth and influence the biological and chemical factors that affect the environment inside the mouth is called the oral microbiota. This microbial ecology is also influenced by the conditions of the person and the mouth, nutrient intake and pH, for example. However, the exact habits the human do that influence this consortium of microbes is not well known. Therefore, researchers at the Department of Odontology at Umeå University, Sweden, devised an experiment to better understand these interactions. 176 consenting participants aged 17 to 21 were gathered from a dental clinic in Umeå. These participants recorded their nutritional intake on a questionnaire, including things like sugar and alcohol. Saliva was collected from these patients to analyze their oral microbiota. These microbes were grown for 48 hours at 37 degrees Celsius and 5% CO2 on plates designed to select for these microorganisms. These plates are known as MSB and LSB agar. Their numbers were determined by counting the number of colony-forming units. DNA was extracted at the laboratory of the Swedish dental clinic. A genomic DNA kit, Genolute bacterial genomic DNA kit to be specific, was used to extract genomic DNA from the saliva samples. A spectrophotometer, Nanodrop 1000 spectrophotometer, was used to determine the quality of the DNA. A fluorometer, Qubit 4 fluorometer, was used to determine the quantity of this DNA. Sterile water and a consortium of known bacteria were used as controls. Statistical tests were also performed to determine microbe quantity and quality for each participant. The average nutritional intake found was 1,856 calories per day. This intake was composed of 40% carbohydrates, of which 5.8% was sucrose. The tests found 127 different bacterial genera. Prevalent taxa include Streptococcus, Prevotella, Haemophilus, Rothia, and Actinomyces. The authors' findings support the ecological plaque hypothesis that entails a shift of the oral microbiota toward a more acid-tolerant bacterial species in an environment of low pH, such as post-consumption of sugar. However, sugar intake monitoring may have been affected by performing this study in a dentist's office, since participants would know that they should not eat sugar beforehand, as it is bad for teeth. That's all for today. Thanks so much, and stay safe out there.